All right, welcome back for part 2. Where we stopped was thinking about 3 patterns of natural selection that we see in phenotype changes over time. And so if we all think about polygenic traits that create a range of phenotypes, we usually get this nice bell curve where there's not many extremes and everybody else is in the middle. If we see that bell curve changing over time, it's showing that the population has changed over time. So the first pattern we'll talk about is stabilizing, and this is when the intermediate, the middle, the most average phenotype is favored. And the other part of this is the extreme phenotypes at each end, the tails, are selected against. Okay, so average is good. Okay, this actually is the most common form of selection because as long as the environment's pretty stable, everything adjusts and adapts to that environment. So a lot of times for stabilizing selection, we think about birth size, even human birth weight. It's stabilizing over time, right? We're not going to wiggle that too much because there's kind of the sweet spot between, 5, 7, 8 pounds. It's a pretty narrow range. Now, obviously, we have little bitty preemies that are born, and we always have some big ones that are born every year that are almost record breakers. But for the most part, we have a very tall and skinny bell curve. This one is about a type of starling. These are the kind of starlings we have around here as well. The clutch size is the number of eggs. Okay? And so if you think about there being kind of a wide bell curve of this where you could have 1, 2, 3, all the way up to 10, 11, 12 eggs, really over time we've seen, and there's actually research for this, that over time that clutch size is going to kind of tighten around just about 4 or 5. That's the sweet spot, right? That, sure, there's still some nests that have less than 4, and there's still some nests that have less than 5, but more than extreme, they're very, very rare, okay? Versus when we think about a normal bell-shaped curve, which means, you could go out and find nests of 3 and 2. Obviously, they wouldn't be as common. Well, now they're almost rare. Directional selection, I think, is pretty easy to understand as well, and that's because one of the extreme phenotypes is favored. Okay, and so this kind of shifts the bell curve to the right or to the left. Okay, this is usually seen when the environment is changing, right? The environment is I shouldn't say it's constantly changing because for the most part, besides this global climate change where we're getting warmer, especially in the tropics, the environment is pretty stable. In the water, the environment is fairly stable. And so when there is a large change in the environment, we're going to see that bell curve shift one side or the other, to the left or to the right to favor those extremes. Some examples of this, and we'll see some pictures of this coming up. Industrial melanism is the classic example of the peppered moths in the Industrial Revolution starting in England in the 1800s. There was so much soot from the coal-burning factories that it actually darkened the trees and the ground and the rock surfaces of the area, and therefore populations, because they now didn't have camouflage for this darker coloration, started shifting towards darker phenotypes over time. And again, individuals didn't change, but the individuals that normally came out a little darker in the next generation had a lot more of an advantage in this new environment. We see this in bacteria as they become resistant to antibiotics. And again, they don't acquire this resistance from the environment. It's just a matter of sometimes a random mutation occurs and they're okay with, they don't feel the effects. They don't get killed by penicillin or some of the other antibiotics. And over time, we see that change, that shift. We also see it with the resistance of pesticides and insects over time, having to have stronger and stronger pesticides to continue to kill off the pests. We see that One of the reasons we don't have malaria in the United States is we killed most of the mosquitoes after the Civil War, and DDT was one of the best used chemicals up through the 60s and 70s to kill mosquitoes that carried malaria. Well, it killed all the mosquitoes, but especially the ones that carried malaria. Well, what we're finding is there's actually a very small percent that have become resistant, meaning sure enough, they go ahead and have all their eggs, and yet there's just a few that survive, and that's all we need. Of course, we can look into the ancient past, and we see how the horse family have changed from forest conditions, and then their body has adapted to more of a grassland condition. So I've got some pictures to look at this a little bit. You can think about a little separated stream where there's two little pools on each side, and we can actually see that by taking away predators, we see that other colorations can be favored because there's no pressure to get rid of that coloration. If these bright blue ones, they don't have any predators. They've kind of set up the pond this way. So there's no predators in one area, and so therefore all the different colors can survive. Whereas if they're in an environment with high predation, only colors that are going to allow for camouflage will survive. Disruptive selection is when both or more than both of the extreme phenotypes are favored, and the intermediate is actually not favored. Okay, and this one's a little more rare, but we do have the British land snails. They're kind of found all throughout Britain, and sure enough, if you find them in fields, they're going to have dark shells, and they don't have any banding patterns. And in the forests, you're going to see mostly snails with light banding patterns. So they're still the same species, but they have different colorations based on their environment. And that kind of makes sense, right? It's pretty shells. So that they can blend in. So if you thought that at one point this was one type of snail population and they had changes from light banding to dark banding, over time that middle was selected against. It was more beneficial, maybe because of camouflage, maybe because of heat regulation. Having one or the other was beneficial. And we could imagine that if these continued to separate, if that disrupted those two types of snails from reproducing to one another, that's kind of the start of speciation. But we'll get there next time. So the thing is, and we've said this a few times, so I don't feel like I have to beat this over your head too much. Even though we look outside and we think, wow, all those organisms are just perfectly suited for their environment, that's not really how natural selection works. It's got to start with the variation that's already present. And then from there, natural selection takes over. So we're always constrained by the variations that are in the population at this moment. Okay? So if we think, oh man, it'd be awesome if I could sprout feathers to fly. Well, there'd have to be someone in the population, and more than one, more than likely, that had that variation to begin with. Okay, we can't induce those variations based on our desires, right? Same with our little airplanes, right? In our lab that we did. It's not that we could just add in whatever variation we wanted once we started the simulation. We had to go with the variation that was already present in that founding populations. So what we actually see is imperfect organisms. If we look deeper, we can actually see that we're not perfect for the environment. There are imperfections that come along with the ride because there's compromises that have to happen because we're not designing perfect organisms here, right? We're not saying, oh, in order to be successful in this environment, you have to have these 10 traits. No, they have to come from the population that's already existing. Okay? So, for instance, we do walk on two hands. We've gone from 4 being quadrupedal, being on like most animals are, that walk on their what we would call hands and feet, and humans do not, along with some of the other primates. But our spine takes a beating because of that, right? Still, a lot of our muscles and our bones are still equipped for having us be on all fours, which we don't walk that way. So again, we're not perfect for our environment either. That's why we have issues with our backs and other things. So when we think about a population, it's always going to have some genotypic variation. So sometimes the phenotypic variations are very slight, but when we look into the genetics, there are differences. Just inherently, we talked about how those accumulate. However, if we have a very limited population, sometimes those environmental conditions, if they change, none of them are good, right? So we need to make sure that there's this constant movement of genes. And so this happens in a few ways. First of all, and we've talked about these when we were in the cell division chapters in protein synthesis, mutations are happening all the time, whether induced by errors errors in DNA replication or chemicals in the environment. There's always mutations occurring. Some of them get passed down, some of them don't. We also have the recombination idea of when an independent assortment, right? That when those homologous chromosomes line up in meiosis, we never know which ones are going to get separated out into individual gametes. Then, of course, when we add one gamete from one parent with another gamete from another parent, we have a bunch of new combinations. So we do usually have this mixing of genes. We also have this idea of gene flow, and we're going to go into more information about that probably in the next lecture as I'm running out of time. But how certain genes get passed on and spread throughout a geographic area. And so here's the thing. While natural selection favors certain phenotypes, still there's others that are good enough. Sometimes instead of saying natural selection is survival of the fittest, sometimes we need to say it's survival of the good enough. And if you watch some of the videos, they may say that phrase as well. So the thing is, in the future, it's not going to that we're all going to look the same and we're all going to have the same genotypes. There's always going to be ones that are just good enough to squeak by and survive for one more generation, and then one more generation, and then one more generation. We'll talk about diploidy and what it means to have an advantage of being heterozygous, having both a dominant and a recessive trait. So again, this lecture we're kind of putting together the genetic side because we are thinking outside the world of Darwin. We can think about how genes are passed down and those cellular processes that occur. Okay, so I'm going to pause for right now, and we'll have another segment coming up next.